Thank you very much. Not everyone wants to have a pathologist stand up and talk about potential diseases in your crops, but hopefully this is a good informative presentation. And as Laurie mentioned, I will be covering some basic aspects of blackleg, and because I work in Washington State University, I'll be putting some emphasis on the situation in Washington State and the quarantine that exists in Washington State for blackleg, and Cindy will proceed with looking at the situation in the Willamette Valley in particular, and the Oregon ruling, and so on. And as a graduate student who's working with me on some Brassica diseases, Shannon Carmody, standing at the back, has a survey she's put together to help raise awareness of some of these seed-borne pathogens on brassicas, and we greatly appreciate it if you'd be willing to fill out the survey now before we get too far in there, because some of the information we presented will change your response potentially to the survey. And so this is a way to gauge people's awareness of both the diseases as well as the ruling that currently and may change, be modified in Oregon and Washington. So thank you very much for filling that out. Okay. With that, I'll move forward. As you all know, we have many different types of brassicas grown in the Pacific Northwest. We have cover crops, we have vegetable seed crops, we have oilseed crops, we have oilseed seed crops, we have seed crops for cover crops, we have fresh market and processing vegetables, green manure crops. There's a wide range of brassicas, and unfortunately the disease we're talking about today doesn't discriminate within the Brassicaceae. It tends to be able to infect almost anything in the family Brassicaceae. So this is one of the difficulties. Many of our farming operations, there isn't just one type of brassica crop, and there's also brassica weeds that can contribute to the importance of this disease, particularly on the maritime side of the Cascade Mountains, where we can do year-round production, and we have year-round brassica weeds potentially in some of our farms. For those of you growing seed, we have a lot of very high-value specialty vegetable seed crops in the Pacific Northwest. I list here some of those in northwestern Washington and the Willamette Valley on the cool maritime side of the mountains that have been grown since the late 1800s. And then in the Columbia Basin of central Washington and central Oregon, a number of these vegetable seed crops. And all the ones underlined in red are in the Brassicaceae. So we have a tremendously important brassica vegetable seed industry in the Northwest, both sides of the mountains. These cumulatively may not seem to be that many acres if you compare with, for example, canola or potatoes or some of the other crops, but they are extremely high value crops. Can range anywhere from $1,000 to over $10,000 per acre. They produce a very significant percent of both the U.S. and the global seed supply of these vegetables because of the unique climatic conditions required for producing high quality seed. We have many other crucifers, as I mentioned. We have a lot of canola production. In Washington State in 2014, we had over 50,000 acres of cover crops in the Brassica family alone. We have a lot of forages and so on. So this leads to why do we worry about blackleg? And I'm going to first give a bit of perspective from Washington State growers. Some of you may be aware that 6 counties in the northwest corner of Washington State, parts of those 6 counties, have been under a brassica quarantine since 2006. So this was before the current concern about blackleg that's shown up in the Willamette Valley. Why is there a quarantine in this area of Washington State? Well, those of you who were around in the 60s and 70s, or who have, a few of you raised your hands. Thanks, Gary. At least one here. Frank maybe was around the 60s. I just squeaked in there. I wasn't looking at brassicas yet, but there was an outbreak of blackleg in brassica crops, vegetable crops, in the Midwest and East Coast, and it was all tied to seed produced in the Northwest. And it was very clear that the Northwest had been a key source of infection that led to these outbreaks in states that have very, very conducive climates, humidity and rainstorms and so on. And it had a big impact on the vegetable seed industry in the Northwest. And so the vegetable seed industry in Washington, Oregon has had a significant impact from this disease and a great awareness historically that's carried through to the current generation of the seed industry and the vegetable side of things about being nervous about blackleg. Making sure we bring in clean stock seed when we're planting seed crops, making sure we're testing seed and making sure we're not bringing in it on seed. In the mid-2000s, Washington State, there was a big push for introducing biofuels into operations of state vehicles, including the ferries in the Puget Sound, which use a tremendous amount of fuel. And the state government wanted to subsidize biofuel crops grown in-state to support the use of biofuels for running state vehicles. And the Puget Sound Seed Growers Association up in the northwest corner of the state was quite concerned about the influx of poor quality seed, particularly infected seed. And they requested the implementation of a quarantine not to prevent the planting of biofuel crops, but to prevent the planting of infected seed that might put the vegetable seed industry at risk of a reoccurrence of what happened in the early 70s. And so this is a picture of one of our seed growers, Tom Holbert from Holbert Farms in the Skagit Valley, his family has grown seed crops for now 3 generations and really have a strong history and a recognition of the importance of high-value, clean seed production in order to maintain that industry there. So blackleg was obviously very much on the radar, and another disease that falls currently under the quarantine for that part of Washington state is black rot. It's a bacterial disease, Xanthomonas campestris, very readily seed-borne and very readily seed-transmitted, and the seed and vegetable seed industry has a zero tolerance for that pathogen as well. So the quarantine for northwest corner of Washington state exists for both of these diseases. In addition, any brassica seed planted in those counties must be tested for dormant seed. Now, this is not a disease-related issue, but if you introduce seed with a lot of dormant seed in there, you're going to have volunteers for a long time to come. And brassicas, crucifers are notorious for volunteer seed and dormancy in seed lots. And if you're planting, say, an oil seed crop or a cover crop and there's a bunch of dormant seed, you can have volunteers for many years to come that now present a cross-pollination risk for seed growers. So this is another requirement for crucifer seed planted in those counties must be tested for dormancy. And if there's dormant seed, that seed cannot be planted. And again, it's not to prevent people from having diverse types of brassica production, but it's to protect the high-value vegetable seed industry. So that quarantine was implemented in January 2006. And for those of you who might work in that area or seed companies selling seed into that area, just so you know, the 6 counties that are partially covered by this, so Clallam Island, Lewis, Skagit, Snohomish, and Whatcom County. It covers not just seed, but any material that might be planted. It covers crop residues from crucifers as well. If you are going to be planting any form of brassica seed in this regulated area, you need to file a notification with a compliance form with WSDA seed program. You have to prove that you've tested the seed for these 2 regulated pathogens and that you've tested the seed for dormancy. And if you want more information, it's WAC Rule 16-301-510. If your seed lot tests positive, it doesn't mean you may not plant the seed lot. It means you have to then treat the seed and retest it to make sure you've eradicated infection on the seed lot. So it doesn't completely rule it out, but you do have to go through that testing and treating and then retesting. There are a few exemptions. This was a big form of discussion during the development of this ruling. What do you do with research trial grounds or breeding programs where you've got lots and lots of different seed lots with very, very small amounts of seed, and you can't test 1,000 seed or 30,000 seed, which is the size they want you to test for black rot. So these exemptions were built in, and our research center in Mount Vernon, Washington is smack bang in the middle of the seed production area where the seed crops are isolated and pinned and so on. And we, despite the fact that we potentially could fall under this exemption, we have mandated at our research center that anyone planting crucifers must follow the rules of testing and treating and so on, because we do not want to be the typhoid Mary, the typhoid John of the seed production area. So there are these minor exemptions for Washington State. So this quarantine was implemented in 2006, and so far we have not yet found blackleg in northwestern Washington. Now, you could say, well, maybe it would have remained free of blackleg even without this quarantine, but it was a proactive measure to try and prevent a situation reoccurring that happened in the 1970s. Now, unfortunately, as Cindy will explain in more detail in her part of this, the Willamette Valley has not remained blackleg free. In 2014, Cindy has been surveying crucifer seed crops in the Willamette Valley since 2005 annually, and in 2014, Cindy found blackleg in multiple sites, many sites across the Willamette Valley. At the same time that Cindy found this, I also received a phone call from one of the growers in the Willamette Valley saying, I think I have blackleg, and Cindy and I both independently looked at samples from this farm and confirmed, yes, definitely was blackleg. We got isolates, we tested them, they were pathogenic. The grower followed all the recommendations to do the appropriate steps following that notification. And Cindy will give you a bit more about everything if she did during that year of finding this. But obviously this was quite a concern because by September of 2014, Cindy had found blackleg in 43 out of 61 sites that she looked at in the Willamette Valley. Now this was not a case of Cindy not having noticed it the years before because she had been going out there and looking at crucifer crops since 2005. So clearly something had happened in a relatively short time to allow a fairly widespread introduction of blackleg across the Willamette Valley. And this was quite disconcerting. A year ago from now, a year ago and a day, Shannon and I met with Cindy Oakham to look at some of these crops where blackleg had shown up. This is a picture of a canola seed crop, a canola crop, winter canola, in the Willamette Valley. And there's a picture of Shannon and Cindy in the field. And it looks pretty healthy overall if you look at it just sort of on the scale of having a casual conversation in the field. But if you get up close to those leaves, you see all the leaf spots, the lesions with the little black fruiting bodies of the fungus. There's 100% incidence of blackleg in that field. You notice it's rainy. It's about 40 degrees. Optimal, ideal for blackleg. We walked to the field immediately adjacent to this field, and the field adjacent had been planted in canola the previous season. And we looked at the residues that were sitting on the soil surface. And what you see in this picture is a piece of stem tissue, right around the crown. There's a reason it's called blackleg. Okay, it's the leg of the stalk. And all those black little bumpy fruiting structures are the sexual stage of the fungus forming on the residues exposed on the soil surface, releasing the sexual stage spores, what are called ascospores, into the air that are now wind dispersed. The stage you see on the leaves is splash dispersed, so it results in localized spread within a field. The stage you see on the residues that forms in the winter is the sexual stage that is airborne, potentially far greater distances. Now you're talking field to field spread, not just within field spread. Not a pretty picture. And Cindy had said it wasn't pretty, and so we got to see this firsthand. The same day we drove over to a turnip seed crop. This is Shannon's in a turnip seed crop. It's a 40-acre seed crop for cover crop turnips. 100% incidence of blackleg. Here's a picture of a leaf with classic blackleg symptoms. We also found white leaf spot and light leaf spot in that field. And Cindy will give you a bit more information on that as well. So this really was a case of going from almost never seeing this disease to seeing fields 100% infected, at least in the case of these more highly susceptible brassicas. So how did this happen? What on earth allowed the situation to develop? We can hypothesize and we speculate to some degree, but looking back at the history and the situation and Washington situation, Oregon situation, and what on earth might have happened. So prior to Oregon House Bill 2427 being implemented in 2009, Oregon had these rapeseed production districts that were set in the 1990s. And they were set for multiple purposes. One is to avoid cross-pollination of seed crops, including canola when you want true genetic type of your seed, minimize the risk of volunteers, and minimize the risk of blackleg. And the rules required that any crucifer seed lots planted in these districts must be tested and treated for formolingum, the cause of blackleg, before planting. The field locations had to be pinned, and you could not grow a crucifer in the same field for more than 2 out of 5 years. Again, trying to break that green bridge. If you did have infection, the inoculum would die off over time unless you had this green bridge sequential planting of crucifers. You had to control volunteers. You had to transport your seed in a manner that the seed didn't escape from the truck or the train or whatever you were using to transport it. You had to clean your equipment before leaving fields. Very similar rules applied to radish, Raphanus. It's in the Brassicaceae, but it's a different genus. It can get blackleg. And many of you may recall the conversations that were going back and forth in the Willamette Valley in the mid-2000s about the interest in having canola grown in the Willamette Valley. And it was very controversial. The Specialty Seed Growers Association in the Willamette Valley did not want canola brought in because of this concern about the risk of diseases like blackleg and other concerns. In 2009, House Bill 2427 was implemented to facilitate experimental plantings of canolas on limited acreage in the Willamette Valley. And this is, again, to test the ability for canola to coexist in the Willamette Valley with some of these specialty seed crops. And for some reason, when that House bill was rewritten to facilitate this experimental planting, the requirement to test and treat for blackleg was removed. I asked Cindy how that happened. She said there were no pathologists on the panel. I don't know if that's, I'll just put it there. This is, I think, to me, one of the biggest mistakes that happened is that removal of the requirement to make sure seed planted was coming in without blackleg. At the same time, in the mid-2000s, there was a widespread increase in planting of crucifer cover crops in the Willamette Valley. And also, canola was approved in some of these experimental areas. Now, we're not going to prove what the exact source of introduction was, but the fact that Cindy found it on so many sites in such a short time suggests there were multiple introductions in a fairly short period, not just on one farm or in one field, because Cindy's found it from the North Willamette Valley down to the South Willamette Valley. So ODA immediately responded because of the recognition of the seriousness of the situation. A temporary rule was implemented. This temporary rule, Nancy Osterbauer from the Oregon Department of Ag has been a key player involved in helping develop these temporary rules. There's her contact information if you find more information directly from ODA. It covered most of the genera in the crucifer family, or the Brassicaceae. Again, there was a small exemption for prepackaged seed lots or transplants simply for home use, not for commercial use. Seed lots had to be accompanied by a certificate for blackleg testing and all be treated in an improved manner for blackleg. And this included transplants. They re-implemented as part of this temporary rule many of the practices that were required under the previous rules before House Bill 2427 was implemented. So again, you couldn't have brassicas more than 2 out of every 5 years in the same field. You had to control volunteers. You had to cover your material that you were shipping in trucks or rails, and so on. It also required seed dealers keep records of the seed lots they sold to make sure they met this compliance of blackleg testing. So that emergency rule, the discovery of blackleg occurred in May of 2014, if I recall correctly, Cindy. At least I received a phone call in May of 2014. By June 18th, the emergency rule had been proposed. People got together. That law became effective by July 7th, a pretty quick action in recognition of the seriousness of the situation. The permanent rule became effective in pretty much a year ago, January 13th of 2015. It includes 2 species of the blackleg pathogen. You've heard me say Fomalingum. Fomalingum is the asexual stage of the fungus, and there's a sexual stage, which is the genus is Leptosphaeria, and there's 2 species of Leptosphaeria, maculans and biglobosa. Some people say biglobosa. So just so you're wondering why all these names are associated with blackleg, that's kind of a quick summary of why. And one really good thing about this ODA rule, and I think it's critical for those of you working in Oregon, and Cindy will go on more about this, is that there's a mandatory review every 2 years of these rules to see if they need to be modified. So this is where you, as people impacted by this rule, need to give feedback to folks like ODA and Cindy and others. If the rule's not working, you need to give that feedback, because this rule is no good if no one can follow it. It needs to be a rule that can be implemented and can be adhered to, is realistic. So any crucifer seed under this rule that's planted in the Willamette Valley must be tested for blackleg and treated, even if the test comes back negative. Okay, so that's a big difference from the Washington rule, but it's what currently is in existence for ODA. A quick little background. I mentioned that there's 2 stages to this fungus. The asexual stage, which produces those little black fruiting bodies called pycnidia, that are splash dispersed. The cnidia that it produces are splash dispersed. And then there's the sexual stage, Leptosphaeria maculans or Leptosphaeria biglobosa, which produces, again, these black fruiting structures. They tend to be quite a bit bigger than the pycnidia. They form on those crop residues, and they're airborne. This is a life cycle drawing or disease cycle drawing from Bridgewald, and you can see, in this case, you start off with infected seed. If you start off with infected seed and plant it out, it's very readily seed transmitted. So the risk if you have infected seed is extremely high that you're going to have seed transmission, because you have to have moisture to grow seedlings, and that moisture is conducive to seed transmission. And you can get the disease cycle repeating itself multiple times in the season because it's splash dispersed, and it doesn't take very long once you inoculate for pycnidia to form on those leaves. Here you can see the pycnidial stage, the splash dispersed spurs. And when infection occurs on the more woody type of stem tissue, particularly in seed crops where you've got big stem pieces or racemes, and during the fall it'll start forming the sexual stage, and these structures are lined with thousands and thousands of sacs, and each of those sacs has 8 of the ascospores, and they literally shot out of the fruiting structure like cannonballs and picked up in any kind of breeze, air movement, and blown around. So that's where they become higher risk for moving around. Okay. So what do we do about blackleg? Unfortunately, as I mentioned at the start of the presentation, almost anything in the Brassicaceae is a potential host. We have found it on a number of weed species. Cindy's found it on a number of weed species. I say we are not in Washington, but I've been receiving samples from Idaho. We found it in things like Jim Hill mustard, which is extremely common out on the dryland area of Idaho. And according to the literature, it can survive about 4 years on seed. We were asked in my program to start looking at seed treatments a number of years ago. We couldn't find any infected seed. The disease had not been on the radar since about the mid-70s, when people had figured out the issues related to the outbreak in the seed production area, and all these practices had been implemented and seed treatments identified. It almost fell off the radar as far as causing any significant impact. And we couldn't find infected seeds, so we actually had to generate infected seed lots over a 16-month period by growing brassicas in the greenhouse and inoculating the pods when they were, and trying to generate somewhat naturally infected seed. Because it tends to die off on seed over an extended period of storage. It can survive at least 3 years on crop residues unless those residues are buried and are breaking down. So residues exposed on the soil surface, especially woody tissue that will survive over the winter through to the next year, are very high risk for producing that airily dispersed stage. So you have splash dispersal of the asexual stage, you have potential movement in running water, splashing water, you have the airborne dispersal, you have the potential to move it on seed or transplants if they're infected, and that gelatinous matrix that the spores come out of in the asexual stage, it can stick to your body if you're walking through the field and the plants are wet. It can stick to machinery if you're walking through the fields. If you think about a cabbage seed crop coming through out of the winter, and sometimes those heads have to be cut open if the wrapper leaves are too tight, they have to be staked, they have to be tied, and inevitably at some point on some of those operations the crop's going to be wet. There's a risk of potentially moving it around. And it's favored by wet and cool. So there's a big concern that we have wet and cool a long time of the year here in the maritime Pacific Northwest. So how do you manage it, given what we've just covered on the general basics of the disease? So the recommendation is a 4-year crop rotation at least, which is difficult if you have a very diversified operation with a lot of different types of brassicas, or you have cover crop brassicas in your cycle. You have sequential plantings of brassicas. This becomes very tedious to, or maybe unrealistic to really be able to implement. So a lot of these management recommendations I'm going to show on this slide are the theory on paper. Then there's the reality of a producer and what they have to go through. Avoid wetting transplants. Well, if it's dry and your ground's dry, you're going to have to water. And most people aren't using drip irrigation for production of brassicas. So this is a reality of trying to follow this recommendation. Isolating your crucifer crops. Now, if you're a seed grower, you're going to do that to prevent cross-pollination. If you're not a seed grower and you're planting sequentially to meet your fresh market or your CSA or other market demand, this is a difficult process. If you're a canola grower out on the dryland area and you've got 1,000 acres of canola, where do we talk about isolation? Again, this is easier to write on paper than it is to fulfill in reality in some situations. Site selection can be important in the context if you have really good airflow across the field, or if you're in a tight corner where there's tall trees and you minimize airflow, there's going to be a much longer periods of high humidity, duration of leaf wetness of the canopy, and more conducive for the disease. There are some cultivars with resistance. For example, in canola, they've developed cultivars with resistance to certain strains or certain races of the pathogen. In vegetables, there's not nearly as much work done on breeding for resistance because it hasn't been the most critical disease to deal with in vegetables for a long time, but this may change. And this is an easy one to write on paper. Control your crucifer weeds, and it's a lot harder to do in our climate where weeds can grow year-round, particularly those in the crucifer family. Same thing about avoid working in your field when it's wet. Let's leave it there. Inspection and roguing. This is really important, and as Cindy and I have had to deal with a lot of samples coming into our labs and questions, if you're not sure what you're looking at, get help. get someone to look at it and help verify if you really are dealing with blackleg or not. There are a bunch of look-alike diseases out there, so it's important to get clarification. Sanitation, this is really critical, particularly in organic production circumstances. This is really, really important. Getting rid of infected material, roguing out plants you suspect are infected. For those of you who are dealing with conventional production, and I know this is an organic conference, but it's important to mention here, some operations are mixed organic and conventional. There are fungicides registered that are highly effective against blackleg, and they can be used very effectively if the timing is done correctly and obviously follow label regulations. There aren't good organically approved fungicides for use in a foliar manner against this disease, unfortunately, which is why it's really critical to follow these other recommendations. And probably one of the most important is to incorporate crop residues as soon as possible after harvest. The longer those residues stick around on the soil in the fall, going through the winter, the longer opportunity you have for that sexual stage to form. Okay. And then, of course, don't plant infected seed. Really, really important from organic seed production perspective. Do everything you can to make sure that the seed you're planting is clean. If you're purchasing it from someone else, find out if the seed lot was tested. If it wasn't tested, you get it tested. And in the case of ODA, now there's a requirement for treating. In Washington State, if it tests negative, you don't have to treat. Hot water treatment can be highly effective. It has to be done very precisely, both in terms of temperature and duration, in terms of consistency of that temperature over the duration, as well as cooling that seed immediately after heating it and very thoroughly drying it to prevent a loss in shelf life and vigor and so on. There are a number of fungicides available for the conventional side of brassicas. What's most important of all as you look through this long list is that if one grower follows all the recommendations but the neighbor doesn't, you still both have a problem. So this is why we're really kind of going around like preachers about blackleg and the importance of regional understanding, education about this disease, and making sure we're all doing what we can to minimize movement of this disease. For the seed treatment side, and this is, I know, a very big issue, particularly with Oregon law requiring both testing and treating. In the organic side is that hot water treatment can be very, very effective. It has to be done very precisely. And Cindy will talk a bit more about it. She's also putting on some hot water treatment workshops this month in the Willamette Valley. If you want to find out more, Cindy will give you more details. For the conventional growers, I just want to mention, although this is an organic conference, there are a lot of products registered for use on brassica seed, but not all treatments are equal when it comes to efficacy against blackleg. And I think that's one of the concerns I've seen, for example, in the canola industry. Almost all canola growers that are doing conventional canola production are using seed treatments, but they're not using the most effective products. So I've listed them here in the seed treatment work we did over 3 years. The top dark orange is the most effective of the products we evaluated, going down to the least effective. They all have some efficacy, but there's a wide range in efficacy. So even in the conventional industry, although they can slap a fungicide on the seed, it may not be that effective if it's not the right choice. And of course, we've talked a little bit about foliar fungicides, which is really relevant to conventional growers, not to organic growers. And because I've mentioned pesticides on this presentation, whether it's organic or not, you're responsible for following the law and reading the label so that you don't sue me if WSDA or ODA doesn't come and sue me. That's all the slide says. Okay. So coming back to the Washington perspective, and I realize most of you may be from Oregon, but I want to bring this up because it leads into Cindy's part of the presentation. We have a quarantine in Washington State that protects part of 6 counties. Does it suffice? What about the rest of the state? Given what's happened in Willamette Valley, do we need to look at additional protections in Washington State? And this kind of sets, as I said, the precedence for what Cindy will be following up with. In November of 2014, I was speaking to the oilseed growers group over in Eastern Washington about this disease because they had heard about blackleg showing up. And they're very aware of this because it's the #1 problem for canola production in Canada. And one of the growers in the audience said to me after I was done, "Would you mind coming out to the parking lot with me? I have residues in the back of my pickup." And sure enough, they had blackleg. And this came from a rapeseed seed crop in Lewiston, Idaho, about 10 miles from the Washington border. And it was blackleg. And we took isolates off these residues and we inoculated cabbage, as you can see in the bottom picture here. And we killed cabbage with their isolates. So they were very aggressive or virulent. A year ago, from March through April, I started receiving phone calls and emails from canola growers in Idaho, west central Idaho. We're seeing these spots in our winter canola, the leaves of the young plants. We think it's blackleg. The picture certainly looked suspect. So I said, well, you're welcome to send them to me, or you can find an Idaho pathologist. And I think because I had worked with blackleg, they decided I was the contact person. So I received samples from about 20 different fields of overwintered canola or canola residues in that area of Idaho, and they all had blackleg, including Jim Hill mustard, the Cecimbrium species. So they considered it a non-issue in Idaho, and now it's all over that area of the Camas Prairie. And Don Hornick from OSU in Hermiston area identified blackleg in 2 overwintered canola crops that under center pivot irrigation in Umatilla County, which is just south of the Washington border. So here we have Washington State, and all those yellow pins represent areas we now know blackleg has been reported in the last 2 to 5 years. And these are the areas of Washington that we have very critical crucifer seed production, either for vegetables or for cover crops or for canola. Do we have enough protection for these areas? It's getting awfully close. As one field was an experimental canola trial ground, cultivar trial ground in Moscow, Idaho, which is 5 miles from the border of Washington. So we're a little concerned in Washington. All of these isolates we've been collecting, and Cindy is now also working with Dr. Delenta Fernando at the University of Manitoba. He's been doing a tremendous amount of work on blackleg in canola production in Canada for many years. So we've sent him all these isolates. I think I've sent him over 50 isolates now from the Willamette Valley, from Idaho, and so on. And they come back mostly the Leptosphaerum maculans species, but also some of the Biglobosa species, including from Willamette Valley. What's interesting is the isolates that have come out of the Willamette Valley from vegetable crops have a very different genetic or virulence phenotype, they call them, the isolates that have come off canola in Idaho. So it looks like we've had some very different separate introductions, which makes sense. We're dealing with vegetables versus canola. All the Idaho isolates that came out of canola crops are resembling the Canadian predominant types. So probably came in on canola seed to that area, unlike what we think is happening in a lot of the Willamette Valley area. So what about the Columbia Basin? It is the last blackleg-free area for canola seed production. Most of Canada's seed for canola production comes out of the Columbia Basin. So they're very concerned about keeping that area blackleg-free. What about the dryland areas? We have a tremendous amount of dryland canola and camelina production. So Dr. Tim Powlitz with the USDA and Karen Sowers, a WSU Extension person, have been surveying overwintered canola crops all spring last year, and they did not find any blackleg, which was a huge relief and a big surprise, but we're very happy. And WSDA did a survey of companies that were selling seed east of the Cascade Mountains. And most, but not all, are testing for blackleg. About 10% of the companies that responded said they do not test for blackleg or black rot, and they do not treat the seed. So there's still a potential risk of introduction. This was a year ago. So as a result, WSDA met together with stakeholders and specialists like myself and others to look at whether we should extend the WSDA quarantine to protect counties east of the Cascades. So there's a proposed amendment to the rule so that counties east of the Cascades are now going to be protected against blackleg. Black rot's not part of the equation. They didn't want that in there. The seed has to be tested with an approved blackleg assay. It has to receive a tag. If the mother lot's been tested, so if a company produces a large amount of seed lot and then sells that to other companies, you can get a daughter tag, so to say, so you don't have to retest every daughter lot that's broken out of that mother lot, because it gets really expensive and tedious running all those tests. WSDA still has a situation where if the seed test is negative, you do not have to treat the seed. Unfortunately, last fall, I received a phone call from Tim Powlitz in late summer, early fall, saying he had looked at canola crop residues in 2 fields that he'd gone to, where he had thought those residues were maybe infected with the sexual stage. And sure enough, we did identify Leptosphaeria from those canola crop residues. 1 field in Okanagan County and 1 field in Whitman County. These are dryland counties in the Palouse. But that's the first report of blackleg in Washington State since the 70s. These are pictures of our cabbage seedlings we inoculated. The picture on the right shows the control where we wound and put water on the wound, and you can see the wound heals over, there's no lesion. The middle picture is the seedling we inoculated with what turned out to be a Leptospira maculans. You can see it very quickly kills that seedling. The Leptospira biglobosa isolates, in this case, they colonize that wound, cause a lesion, but they're not nearly as aggressive or virulent as the maculans in this case. So we have a quarantine. We've produced a bulletin that talks about this complexity of all these different types of crucifers that we grow in Washington State, and that's also the case for Oregon. We have a blackleg interest group, and if anyone would like to be on that, Cindy has her own mailing list, I know. And our seed program manager for WSDA, Victor Shaw, has a listserv if anyone would like to keep up to date on the current ruling in Washington State with regards to blackleg. One really important thing, and this applies to, some of Cindy will talk about this too, but we have a lot of really good diagnostic resources available for folks both in Oregon and Washington. I list all of the Pacific Northwest diagnostic labs. If you're not sure what you're dealing with and you want someone to look at it, and it's not Cindy or me, we have Robert Keating in Hermiston, Melody Putnam and Morena here in Corvallis, Jenny at Puyallup, and Rachel, a new diagnostician in Pullman. And the University of Idaho has a new pathologist starting in a diagnostic capacity, James Whitehall. He'll be starting next month in Palmer, Idaho. So it's a really nice network of diagnostic resources if you need help with that. I mentioned how Cindy had also found light and white leaf spot. I just wanted to say that we have a graduate student standing. Shannon, you want to raise your hand? Shannon is funded by the Cliff Bar Foundation Seed Matters Fellowship. And part of her project is to look at light leaf spot and white leaf spot. Cindy's on her committee as well. Cylindrosporum causes a light leaf spot and Pseudocercospirilla causes white leaf spot, to see if these diseases are of any concern for us, particularly when it comes to the seed-borne aspects. Should we worry about them? Cindy and I have, and Shannon have been looking at these diseases to see if we should just ignore them or if we need to worry about them. And this is some data out of the UK that shows 4 diseases that affect rapeseed or canola production. The blue bars represent Phoma blackleg. The red bars represent light leaf spot, Cylindrosporium. The green bars are Sclerotinia, which causes white mold. And the grayish bars or purple bars are Alternaria. And the height of the bar represents the extent of losses caused by these diseases in pounds, millions of pounds, the currency in the UK, from 2005 to 2014. And look at light leaf spot. It's not a good picture. Now, rapeseed is planted on a pretty wide scale across the UK, so that's obviously contributed to the predominance of this disease, but it has become the primary cause of losses in rapeseed in the UK. So we believe that this is why both Cindy and I both have graduate students working with us to look at these diseases in the realm of the Pacific Northwest and what we might be able to do if these become more predominant in our area. So this is Shannon inoculating some cabbage in a closed situation in a greenhouse to try and generate naturally infected seed lots, looking at the potential for seed transmission, whether the treatments we use for blackleg will hold up against this disease. And she's also doing extension outreach work, which is why she's handing out a survey at the start of this session to get an understanding of people's knowledge of these pathogens. So we thank the Cliff Bar again for funding her project and supporting this.